Bismillahirrahmanirrahim. Assalamu alaikum guys. Today our topic is lymph. A typical lymph node structure. A lymph node, its structure and its function. A lymph node or lymph gland is a kidney-shaped organ of the lymphatic system and the adaptive immune system. Lymph nodes are small bean-shaped glands that are located along the lymphatic system, a system of vessels similar to arteries and veins through which lymph fluids travel. Lymph nodes are classified as secondary lymphoid organs, with the primary lymph organ being the thymus gland, tonsils, spleens, and bone marrow. Function of lymph nodes. Lymph nodes work like filters or in our analogy security guards to filter bacteria, viruses, parasites, and other foreign particles, even cancer cells that are brought to the nodes via lymphatic vessels. Lymph nodes also play an important role in fighting against infections. Our second question is number of axillary lymph and their terminating places between 20 and 49 in numbers. They drain lymph vessels from the lateral quadrant of the breast, the superficial lymph vessels from thin walls of the chest and the abdomen above the level of the navel, and the vessels from the upper limb. The axillary lymph nodes arrange into five groups based on their anatomical relations. They drain the wall of the thorax, breast, arm, and upper abdominal wall above the umbilicus. Thank you.
I will tell you about direct pathway and indirect pathway. Cerebral cortex will give input to basal ganglia and get output to basal ganglia. So input and output is kind of related to cerebral cortex. From cerebral cortex through corticostriatal fiber, neurotransmitter will produce known as glutamate will go into stratum part of putamen. From there it would go into globus pallidus internum. Globus pallidus internum will produce high amount of GABA, which inhibits glutamate. From there it would go into ventrolateral nucleus, part of thalamus, and from there it go back into motor area of cerebral cortex through thoracocortical fiber. Same would be happen in indirect pathway, but the difference would be it goes in globus pallidus externum except of internum, and a little different would be more about low amount of releasing of GABA, which will inhibit all process, due to which in indirect pathway cerebral cortex would be inhibited, and in direct pathway as there would be more amount of GABA, it would produce it would heavily impact glutamate. So cerebral cortex would be happy kind of. So globus pallidus internum and externum will produce GABA, and one more thing, substantia nigra will produce dopamine always. So in direct pathway its functions are for initiation of movement and increase cerebral cortex, while in indirect pathway it suppression the movement and kind of decreases cerebral cortex.
Assalamu alaikum. My name is Moawaz Abdul Wasi. I am first year student of OSH International Medical University, Kyrgyzstan. Today my topic of lecture is germ cells, germ layers and neural crust cells. Now I am going to my topic, germ cells. The definition of germ cells are the only cells that live in the body that have half of amount of chromosomes both metastasis and meiosis, and in male produce the gamete sperm. Now going to germ layers. Germ layer, any of three primary cells layer formed in the earliest stage of embryonic development, consists of the ectoderm, endoderm and mesoderm. Now neural crust. Neural crust cells are the multiple end cell induced at the border of neural plate that subsequently migrate throughout the embryo and later differentiate into multiple cells type, contributing to most peripheral nervous system and the craniofacial cartilage and bone as well as ligament and endocrine. Thank you.
Today we discuss about nisal granule and chromatolysis. First of all, we discuss about nisal granules. Nisal granules are very small and they are present in soma or paracaryon. Soma mean body, so they are present around the nucleus in large amount and in cell body. They are present around the nucleus and cell body. So if we discuss about the function of nisal granule, then ribosomes are present in nisal granule. Ribosomes are present. So first function is production or synthesis of protein, and then second function of nisal granule is staining. So that's our nisal granule, our small topic. And then we discuss about chromatolysis, the process of dissolution of nisal granule is called chromatolysis. This nisal granule disappear due to some injury or fatigability of neurons. Due to injury or fatigability of neurons. In simple word, we can say it is in this case nisal granule are disappear in chromatolysis. Then we discuss in neuron, in this case is swell up. Neuron swell up हो जाते हैं. Swell up means सूज जाते हैं. Then if this injury will be recovered or this fatigability will be recovered of neuron, then nisal granule or reappear again. So that's our chromatolysis and nisal granule.
Assalamualaikum. I am going to explain the topic of Schwann cells, and Schwann cells do the myelination in peripheral nervous system. And here I have made the structure of Schwann cells, and they do myelination in peripheral nervous system. This is the structure of neuron, and neuron is a structural and functional unit of nervous system. And you can see that this is a neoplasm of neuron, and in the neoplasm of neuron there is a nucleus, mitochondrial, and pericardium or soma, which is a cell body. And when we talk about the special characteristics of neuron, then these are nasal granules and neurofibrils. And nasal granules, in case of any injury or fatigue in the cell, the nasal granules disappear and the cell is swelled up. And then after the recovery or repair process, the nasal granules reappear. And this is what we call as chromatolysis, that is a special characteristics of neuron. And then when we talk about the myelin sheath, this is done in the peripheral nervous system by Schwann cells and in central nervous system by oligodendrocytes. And in the peripheral nervous system, the difference between two node of appears, it is called as internodal path, and and then the covering of neuron is called as epimysium, and the covering of fasciculus is called as perimysium, and the covering of nerve fiber is called as endomysium. And when there is a degeneration of oligodendrocytes in the central nervous system, it is called as the myelin sheath of oligodendrocytes in the central nervous system is called as multiple sclerosis, and the degeneration of the myelin sheath, which is made by Schwann cells in the peripheral nervous system, it is called as Guillain-Barré syndrome, and it includes the ascending paralysis of lower limb, abdomen and thorax. And in the thorax region, when it came over there, then it caused respiratory arrest of the person and death happens. Thank you.
We are talking about the anterograde flow and the retrograde flow. The flow from the cell body toward the synaptic cavity is called anterograde flow. It is fast flow. The anterograde flow is due to the kinesin. Its speed is about 100 to 400 ml per day. The air is an exogonal polyneuropathy. In the anterograde flow, for example, in anorexia due to the lack of oxygen, the mitochondria can't do the oxidative phosphorylation. Then the microtubules will be disturbed, diabetes mellitus, and the hyperglycemia increase glucose level in the blood. Depolarises the microtubules, as a result of which there is a pathology of glove and stalking pattern in which there is an alternated sensations of pain over the hand and foot. Then we talk about the retrograde flow. The retrograde flow is from the synapse towards the cell body. Its flow is about 60 to 100 ml per day. It is the slow process as compared to the anterograde flow. It is because of dynein.
Assalamu alaikum. Today is my topic myelinogenesis. Myelinogenesis formation the myelin sheet around the neurons in central nervous system as well as peripheral nervous system. In central nervous system, oligodendroid cells, and in peripheral nervous system, Schwann cells from the myelin sheet. This process start at fourth month of intrauterine life and ends second year of age in children. Thank you.
Assalamu alaikum. My name is Abdul Samad. I am a student of OSH IMU. Today my topic is difference between primitive group, primitive and primitive strix. The primitive groove, the primitive bits is continuous with the primitive grooves, and the primitive strix. Primitive strix is the originate from the interior epiblast and appears as an elongating grooves, primitive grooves on the dorsal mid-sagittal surface of the epiblast along the anterior posterior axis and embryo of the embryo.
Assalamu alaikum. Today I will ask Muhammad J.S. Kishta about why neuron is a permanent cell and cannot reproduce itself.
1. Neurons or nerve cell is defined as the structural and functional unit of nervous system. 2. Neuron is similar to any other cell in the body. 3. Neuron does not have centrosome, so it cannot and undergo definition.
Bismillahirrahmanirrahim. Neuroendoderm. Neuroderm, it has at the site as physical support and have live family acid protein and word blood-brain barrier and area posterima chemotrazole zone and reactive glycolysis buffer system for potassium and glycogen. It removed the potassium and glycogen from extracellular fluid and second is the oligodendroglion cell. It made the central nervous system of a zone of myelin shield. It could one oligo covered the 30 fiber nerve and 30 the epidermal cell. It has three types cell present. First of all is the epidermocyte cell. It produces cerebrospinal fluid, and second it a quarter epithelial cell. It is a simple board cell lining of ventricle, fourth ventricle, and central canal, maculae, and cilia. And third is a tiny site. It transport the substance from blood vessel to cerebrospinal fluid. That's all.